In this video, we are going to look at some individual DNA techniques just briefly and in general so that you have an idea of certain kinds of things that biotechnologists and genetic engineers can do with DNA. The first technique we'll look at is called gel electrophoresis. And this is a technique that we can use to separate strands of DNA. So in the last video, we looked at how to clone a gene, and we took a bacterial plasmid and of a certain size, say it was 2,300 base pairs, and we cut that plasmid, and then we inserted a human insulin gene that's about 1,700 base pairs. So there is a distinct size difference between the empty plasmid and the plasmid with the DNA insert. If we were to run these two samples on a DNA gel, we would see the size difference on the gel. So how does this work? We make a gel substance from a substance called agarose, which is just a type of polysaccharide, like a carbohydrate that comes from a type of seaweed. And when we use this gel, we put it in a buffer solution and we connect it to electrodes. And there's a positive end and a negative end. And so remember that DNA, our nucleotides, are made of sugars and phosphates and bases, nitrogenous bases. The phosphate groups are all negatively charged. So if we put DNA into this gel and then turn on the electricity, which way is the DNA going to be attracted to? Which way will it move through the gel? If the DNA is negative, will it move towards the negative end or towards the positive end? Towards the positive end, because opposites attract. So let's have a look at this diagram. Here's our gel apparatus. We have a negative end and a positive end. And the gel is made of agarose, and it contains a buffer solution. Now when the gel is made, it's liquid, and we put a comb in it to make these little indented wells. And then the gel sets into kind of like the consistency of hard jello, actually. And then we can put our DNA into these little wells. And then when we turn on the electricity, the DNA is going to move through this gel substance towards the positive end. So we add our samples, and we will need to compare our DNA samples with a ladder. So a ladder is known fragment lengths. Now what happens as the DNA moves towards the positive end is it starts to separate the DNA based on size. So the smaller DNA fragments are going to move through the gel faster than the larger DNA fragments. So up here, this one might be 4,000 base pairs, and this one might be 2,000 base pairs, and maybe that one's 500. So as they move through the gel, the smaller ones will move faster and they start to separate, and we can see these bands. Now let's suppose over here this was our plasmid sample. Let's say we added our empty plasmid from our cloning experiment the other day, and then we compare it to the sample that we think has our DNA insert. Well, here we could see that these are both the same size, so this one can't contain the DNA insert. If it did, it would have to be up here because it would be bigger. Then we use this dye called dithium bromide, which binds to DNA, and we can look at the bands under UV light. There's different detection mechanisms, but that's what this one here is showing. The next technique I want to look at is called PCR, or polymerase chain reaction. If you remember from our DNA replication video, polymerase is the enzyme that adds complementary nucleotides. So when we're replicating DNA, we use DNA polymerase. When we're transcribing DNA to make RNA, we use RNA polymerase. So polymerase adds complementary nucleotides. So we use PCR, polymerase chain reaction, so that we can amplify fragments of DNA in a thermocycling machine, so that we don't necessarily have to use bacteria. Bacteria are great for cloning genes and copying DNA. However, because they're living organisms, they can sometimes add mutations. So sometimes over time of growing a population of bacteria, eventually the DNA sequence might mutate a little bit. If we use PCR, we can amplify one fragment of DNA quite quickly. So, what are the ingredients that we need to use PCR? We need to have our gene of interest that we want to amplify, or our fragment of DNA. We need to provide the polymerase. In PCR, because it's a thermocycler and the temperature is going to be cycling and changing, going from hot to warm, we need to use a Taq polymerase because it comes from an organism that normally lives in thermal hot springs and it can tolerate the heat. Normally, proteins like enzymes can denature in high heat, so we use a specific enzyme called Taq polymerase. And then we have to have our nucleotides. We have to add our A's and T's and C's and G's. So the polymerase has substrates to add to our DNA sequence. And then lastly, we need to have a primer. A primer, remember from DNA replication, polymerase can't begin copying DNA unless there is a primer to start with. So we create those primer sequences and then the polymerase knows exactly which fragments of DNA to copy. In this diagram, we can see that we are starting with a target DNA sequence. Here is our gene that we want to amplify. Let's suppose it's our insulin gene. And it has a 5' end and a 3' end, and they are anti-parallel just like every DNA fragment. Now, when we replicate DNA in the human body, we use an enzyme called helicase to unwind and open the DNA so that each strand can be copied. In a thermocycler, we don't use helicase, we use heat. And heat will denature or break apart the hydrogen bonds that are holding the double strands of DNA together. So we heat the DNA and the strands will separate. Then we change the temperature again. Now we decrease the temperature. So let's suppose we denature this at about 95 degrees. So that high heat will separate, break the hydrogen bonds. Then we have to decrease that temperature to, say, about 55 degrees so that we can add the primers. Now the DNA primers, they will bind because they have the complementary sequence. And we need to have two. We need to have a primer that starts at the 5' end and a primer that starts at the 3' end. And then we change the temperature again. So Taq polymerase happens to enjoy about 72 to 75 degrees. And then at this temperature, the polymerase will start adding the new complementary nucleotides. And now after one cycle of changing the temperature, we have gone from one DNA fragment to two DNA fragments. But now remember that we're not going to start with one single DNA fragment. We will most likely be starting with thousands of DNA fragments. Then each cycle, we are going to be exponentially increasing the number of DNA fragments. And we do this over and over and over. About 30 cycles will give us a hugely significant number of DNA molecules. So PCR is a really valuable tool for being able to amplify pieces of DNA. Now we also have what's called RT-PCR, which stands for reverse transcriptase PCR. And then we also have RT-PCR that can sometimes mean real-time PCR. So real-time PCR should actually be technically called quantitative PCR. With real-time PCR, we can amplify sequences of DNA using a fluorescent dye, and the machine can then look at the amount of light and basically quantify the amount of DNA that's being produced. I want to focus a little bit more on reverse transcriptase PCR. When we have DNA, a chromosome inside of our cell, and we want to make a messenger RNA molecule from a gene sequence, we use RNA polymerase to copy one of the strands into a messenger RNA molecule. So then remember that when we have messenger RNA in eukaryotic cells, it has to be processed. And we cut out the introns. The introns have to be removed before that messenger RNA molecule can go to a ribosome and translate a protein sequence. So reverse transcriptase PCR is when we take an RNA molecule and we make a DNA molecule. If we look at a processed RNA, remember that in addition to removing the introns, we add a poly A tail and a 5' cap. What we do when we reverse transcribe this, number one, we need to have an enzyme called reverse transcriptase. This enzyme is actually originally isolated from retroviruses, RNA viruses that use this enzyme to make DNA when they infect cells, like HIV is a retrovirus. So we can take this enzyme and we can put it into the PCR machine. So now, instead of starting with a DNA fragment, we're starting with an mRNA, a messenger RNA fragment. So we have our reverse transcriptase, we have our messenger RNA molecule, we need to have our nucleotides, and then we need to have primers. So in this case, when we're going to create DNA from RNA, the primer is going to be a poly T primer so that it will find messenger RNA. And then the poly T will be used as the beginning for the reverse transcriptase, and then we will create a DNA molecule. Now, that DNA molecule that is produced from the messenger RNA does not have introns. So this is called complementary DNA or cDNA. And this is very useful as well because remember how bacteria can't process RNA. They can't remove introns. So actually, when we're cloning our gene and we're using bacteria, we have to use cDNA so that the bacteria doesn't have to process the RNA. So that is a very useful function for RT-PCR. Next, I want to talk about 3 different processes where we can look at either proteins, DNA, or RNA, and then we can look at a whole bunch of those, and then we can transfer them to a filter paper, and then use a probe to find a specific sequence. So if we have a sample, that sample could be either protein, DNA, or RNA. So our proteins are made up of amino acids and form polypeptides. Our DNA and RNA are nucleotides. When we look at protein, it is called a Western blot. When we look at DNA, it is a Southern blot, and RNA is a Northern blot. And so then what we can do is we can take these samples and we can run them on a gel through electrophoresis. This one is showing an agarose gel that we just talked about, which would be useful for RNA and DNA. Protein is run on a polyacrylamide gel electrophoresis, or PAGE electrophoresis. The setup is slightly different, but the concept is the same. So the protein, the DNA, or the RNA is going to run through some kind of a substance. In this case, we're going to focus more on the DNA, running a Southern blot. And just like we talked about in the gel electrophoresis section a second ago, the DNA is going to move towards the positive end, and the smaller fragments are going to move faster. After we've run this whole sample out on the gel, now the next step is mixing that with a buffer and a membrane. It's almost kind of like a coffee filter. It's either nylon or nitrocellulose or some kind of a membrane, and the DNA or the RNA or the protein will transfer into this filter paper. The other thing that we're going to do is we're going to incubate it with a very specific probe. If we wanted to look at very specific DNA sequences, we could use a probe that is either chemiluminescent or radioactive, so we could use X-ray film. There's different ways of detecting the DNA. So now, because let's suppose you took a cheek swab from a human and you digested all of that DNA and ran it on a gel, if you looked at all of it, it would just look like a big giant smear because we have so much DNA. So we incubate that filter paper with a probe so that we only look at the very specific DNA that we want. Now, this is very useful because we're going to come back to that, but I want to explain something else. We have repeating sequences of DNA called VNTRs, or variable number tandem repeats. And all humans have these, all living things have these in our intron sequences. But what's interesting is that every individual, except identical twins, will have different numbers of these repeats. So let's have a look at an example. Here is a typical VNTR sequence in a human. Now, let's suppose Bob has 20 of these repeats in his introns and Sally has 40. Okay, what's going to happen if you take Bob and Sally's DNA and you run it on a gel? So you digest it with a restriction enzyme, so it's all kinds of little pieces and fragments. You run it on the gel, and you make a probe for that VNTR sequence, okay? And then you compare Bob and Sally. Because Sally has longer VNTR sequences, her DNA will show up as a band at closer to the negative side compared to Bob. His DNA fragment is going to be shorter. He only has 20 repeats, so that sequence of DNA is shorter, so it will run faster on the gel, closer towards the positive end. So then you can see that Sally has a certain DNA fragment size based on her VNTR sequences compared to Bob. So how can we use this information? Okay, we can do DNA fingerprinting. This is how we can identify different individuals. So let's look at an example. Suppose we went to a crime scene and you had evidence, blood, there was blood on a weapon, and you've got a victim and then you've got some suspects and there's a bunch of different blood. If we just concentrate on the blood that doesn't match the victim and we compare it to the DNA fragments of, say, 3 suspects, we can figure out whose blood was on the weapon. We take our DNA, we take our cheek swab sample, and we break open the cells, we digest the DNA with restriction enzymes and chop it all up into bits. And then we run it on a gel electrophoresis. And then we use a probe that is specific for VNTRs. There are many different ones, so you could use different probes to look at multiple VNTRs. And then we have our evidence DNA and our 3 suspects. So whose DNA matches? Okay, suspect number 2. He has, or she, has the same VNTR DNA pattern as the evidence. So we know that suspect number 2 and suspect number 3 are different people, and suspect number 2 is the same person as this DNA evidence. We can also use DNA fingerprinting to figure out familial ties and connections. You can see who's a sibling and who's a parent. Okay, so if we look at this example, we can figure out who's the dad. Here we have mom's DNA VNTR sequence. Okay, so mom has a very specific pattern. And remember that mom has 2 alleles for every chromosome, so she has basically 2 copies of everything, and she's going to give 23 of her chromosomes to her baby. So baby Susie is going to have some VNTR sequences that are in common with mom, but not all of them. You can see that Susie is a unique human that is different from mom's pattern, but there are some similarities. So now take a look at this over here and see if you can figure out who is the dad, who also has DNA fragments in common with the baby. Okay, did you see here? Doug is the only one that has DNA fragments in common with the baby. Frank and Ted have no fragments in common with the baby, so it's impossible for them to be the dad. The last thing that I want to talk about is gene therapy. So how can we use all of this DNA information that we have? We know all of these gene sequences. We know where genes are located on chromosomes. And we know of different mutations that cause inherited diseases like sickle cell anemia and cystic fibrosis and Duchenne muscular dystrophy. If we could fix those gene sequences, we would be able to get rid of those diseases. But how do we do it? So the idea with gene therapy is that we take a healthy gene sequence and we insert it into the genome and get rid of the mutated sequence. But how do we do that? Once you have a fully grown human, whether it's a baby or an adult, we have trillions of cells in our body. How do you get the healthy gene sequence to the right location in the genome? You can't just randomly stick DNA into a cell because you don't know where that fragment is going to go. And if it inserts into the wrong place, you could disrupt another gene and just create more mutations. So this has been the problem with gene therapy. It's very difficult to get the right gene into the right cells in the right place so that we can correct mutations. But there is a new technology that has been coming about in the last couple of years, and that is called CRISPR. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And these are a family of DNA sequences that come from bacteria, from prokaryotic organisms. And they are like restriction enzymes. So remember palindromes from our Cloning a Gene video. Restriction enzymes recognize palindrome sequences. Now, this works a little bit differently than restriction enzymes. With CRISPR, there is a specific protein called Cas9, and that is a CRISPR-associated protein 9. And it is a unique protein because it comes along with a guide RNA molecule. Let's have a look at this diagram. Here we have our Cas9 protein, and it contains a guide RNA. This guide RNA molecule will recognize a specific sequence on the DNA. When that sequence is recognized, this protein will cut the DNA and it will delete a sequence of DNA. And then, because we can make this guide RNA sequence very specific, we can use the exact sequence to find the exact location on the DNA to cut out that fragment. We can then insert the correct gene into the correct location. So this is pretty cool because now gene therapy is going to reach new heights. Now we actually have a way of targeting specific genes in a specific location in specific cell types. So clinical trials began with a few different things starting in about 2019. And there have been a lot of successes with things like cervical cancer and blood disorder and a genetic blindness mutation. So this is really exciting because once we can get the correct gene into the correct place, we can remove cells from the person, we can use this guide RNA Cas9 system, put the right gene into the right place. It could be used for multiple conditions, including our Duchenne muscular dystrophy and cystic fibrosis and cancers. So this is an exciting new area of genetic engineering.